Hello, everybody. Thank you for checking out my talk. My name's Dan Baldassarre. I'm an assistant professor at SUNY Oswego, and I'm going to be presenting results here about how urbanization may be affecting northern cardinal color. So to start off, we know that it's tough being a bird in the Anthropocene. A lot of different ways that humans are negatively affecting bird populations. A couple of examples represented here. And we can put some numbers on how severe this situation is. So this recent paper, many of you will be familiar with, suggests we've lost about 3 billion birds in the last 40 years or so. And no matter what category of birds that we look at, with just a few exceptions, the situation is pretty bad. But a little bit less intuitive way that we may be negatively affecting birds is by changing the way they communicate. Okay, so we also are gathering evidence that it's tough to communicate in the face of human activity, especially human noise. Here's a great recent example of white-crowned sparrows adjusting their singing behavior in response to changing human activity and traffic patterns, especially as a result of the COVID shutdown. And there are a lot of other great examples of birds adjusting their singing behavior in different ways to compensate for human noise. We know relatively less about how human activity can affect avian visual signals. So their plumage color, which we know is also a very important signaling modality for birds. So we're going to switch to the visual channel for this talk and think about how humans can affect bird visual signals using this species. So we study this in the Northern Cardinal. They're widespread across the eastern United States. They're a dietary generalist, and they love to be alongside humans. They'll readily breed in urban areas, in people's backyards, etc. So they're a great species in which to ask this question. Does urbanization affect their plumage color, any of their other color patches that they may be using for visual signaling? And there's a good reason to think that it might, because we know that these color patches, both the plumage and the orange bills that these guys sport, are a result of carotenoids. So carotenoids are pigments that birds have to get through their diet. They can't manufacture themselves. These color patches we know are used as signals for various functions in cardinals. And the amount and the type of carotenoids that they get can affect their color. We know that from studying things like mutant cardinals, for example, that show up with weird yellow plumage. And there is some previous research showing that this can be affected by urbanization. So here's a 2010 study that found that urban male cardinals were actually less bright. So out here, these are more urban birds, and higher on the y-axis are less bright birds. And we see this relationship where as you get more urban, the bird's plumage is dulled. So my group is sort of expanding off of this idea, looking at multiple plumage patches in males and females with a little bit updated color quantification techniques to basically ask and test the idea that if urban and rural birds have differential access to carotenoids, they're eating different things in rural and urban areas, that might cause them to differ in color. So we studied these guys at 2 field sites in upstate New York. We've got a rural site and we've got an urban site that's right smack dab in the middle of Syracuse. And we're going to quantify 4 different color patches that may be relevant to their signaling system. In males, we're going to look at back plumage. We're going to look at chest plumage. In males and females, we're going to look at bill color. And then in females, we're going to look at this underwing plumage patch. Female cardinals, one of the cool things about them is they got their own interesting color signals. If you look at the underwing, they have this beautiful pink sort of wash to it. So we're going to look and see how urbanization affects that plumage patch. For our feather samples, we're going to use reflectance spectrometry to get a full spectrum idea of the color that's being reflected by these patches. And then for bills and underwing patches, we have color standardized photos that we analyze to quantify color. In both cases, we're going to run these colors through avian visual models to get an idea of what the colors are as they're perceived by the avian visual system. And we're going to compare urban and rural birds in hue, chroma, and brightness. So here's some basic results. If we look at rural and urban males and we look at their back and chest plumage, we don't see any dramatic differences. So here are some reflectance curves of rural and urban chest feathers up here and rural and urban back feathers. And you'll see the reflectance profiles, the type of light that's reflected off of these patches is very similar in those two comparisons. The reflectance curves sit right on top of one another. And if we put these into an avian color space, we can get our values for hue, chroma, and brightness in a slightly different way. And the avian visual color space we can represent in this tetrahedron. Birds have 4 color cones in their eyes that are sensitive to ultraviolet, short, medium, and long wavelength light. And the angle from the center of this color space out to our color tells us the hue. The length of that vector, so how far away we are from the center of this space, tells us the chroma, which you can also think of as like saturation, sort of how pure is the color. And then a little bit separate from the color space, actually, independent of color, is brightness, which is really just how much light is reflected off of the feather. And so if we put our back and chest color patches in this color space, we see them rural and urban birds sitting right on top of each other. We can't distinguish them. So rural and urban back, rural and urban chest plumage, very similar in this color space. And if we look at our 3 color variables that we're interested in, hue, chroma, and brightness, we don't detect any difference in any linear models. So this is chest down here, this is back. For hue, rural and urban birds similar, same with chroma, same with brightness. But we have a year effect in our data. So we have data collected from 2019 and 2020. In 2019, the chest feathers of these males were the same in all those 3 color variables, including hue, which I'm showing here. But we detect a difference in hue in 2020. So in 2020, urban males actually had redder chest plumage. In this color space here, the color points start to drift apart. Urban birds are actually closer to a red color, and rural birds are closer to an orange color. And we detect a significant difference in hue values in our linear model. So that's interesting. Something's going on with this year. If we go and switch and look at bill color, now we're in a triangular color space because we're analyzing photos and we've lost the UV channel, but still a very similar thing going on here. Urban birds have more reddish bills compared to rural birds. So urban birds in orange, rural birds in green. Urban birds have hue values that are closer to a red color. Rural birds are closer to orange. And again, if we look at hue, this is where we detect a significant difference in bill color. Chroma and brightness, no difference in the bill color between urban and rural males. For females, we don't detect any difference in their bill color between rural and urban birds. Their color points are sitting right on top of each other in the color space. And if we look at our 3 color variables, we don't detect any significant difference. But there is something interesting going on with females. So if we look at that underwing patch that they have, we find a difference in chroma. So urban females have more saturated underwings. They have higher values for chroma. And you can see that here in the color space. Urban females are pink, rural are brown, and the urban females are further away from the origin here. They have higher values of chroma, more saturated, pure pink color to those plumage patches. And that's represented by chroma. If we look at how chroma differs, we find significantly higher values for urban birds. There's no difference in hue or brightness. So it's the same color pink and it's the same brightness, but the saturation is different between the two. So we've got some interesting, sort of tantalizing results here that suggest that urbanization may affect presumably diet, the things that these birds are eating, and their color signals. And this is a little bit different from what some of the previous research in the cardinals has shown. So remember I showed you that figure of urban birds having duller plumage. We don't actually detect any effect on brightness per se, but we do find that our urban birds tend to be redder, which might be slightly different from what some of those previous studies have shown. Also a little bit different from what we see in tits, for example. In Europe, urban tits have duller plumage. But there is some precedent for possibly urban birds of other species having access to probably more invasive honeysuckles, for example, which get more and different carotenoids into their feathers and can cause them to be redder than they would otherwise. So waxwings, flickers, orioles have all shown this. So something similar might be going on with the cardinals if they have more access to invasive shrubs, especially honeysuckles, in urban areas where those plants tend to thrive. But there's still work to be done here to figure out what's going on. We're trying to increase our sample size, do some more population monitoring, and try and eventually figure out if there's any effect of the variation in these signals in these plumages. We want to measure carotenoids that are actually in the feathers to see if those are different between our rural and urban birds, and also trying to figure out what these guys are eating. So using stable isotopes and also doing some metabarcoding of fecal DNA to figure out if urban and rural birds are actually eating different things, or if maybe there are other reasons why their plumage color may differ. So with that, I need to thank my team of undergraduate researchers at Oswego who do the bulk of this work, especially the field work. I couldn't do any of this without them. And acknowledge a couple funding sources. And hopefully you all have some questions in the chat or later in the Q&A. Thanks for tuning in.